Rflp and diversity array technology, draft, dart, single feature polymer. We'll discuss these things. Okay, you know that the structure of the DNA was discovered by the Frederick Mitchell, Rosalind Franklin, and Watson Crick. These are the three important scientists. From their discovery, everything started about molecular biology. Like in 1952, when they discovered the DNA structure, it's not like DNA as genetic material. They told about the structure of the DNA, how nucleotides were arranged. This is actually the model. They used all the information from the previous literature, like Chagraph rule, Herbert Cosell experiments, like how the what is the nucleic acid is contents of, and Chagraph rule, like A is equal to G, pyrimidine is equal to pyrimidine, like purines. By using all the and photograph which is taken by the Wilkinson, and these things they clubbed together and they discovered the helix model. From there, everything is started, even our DNA markers. What we are looking? We are looking at variation. Possible only when we know the structure of the DNA. How the structure, like how it is, how the nucleotides were arranged in the polynucleotide, like in the DNA, and what are all the variation can be possible, like mutations, insertion, deletions. These are the things we need to remember. I think instead of marker, I will start genome organization. So you will get a better idea. My slides are visible.
So the DNA structure is visible.
Actually, today I will start a genomics class. A genome, what is genome organization, so that you will get better understanding. If I keep on telling what is repetitive DNA, what is genome organization, again it's a repetitive thing. Today I will cover genome organization. Now,
Yes,
this
sir.
is visible.
Yes, sir. Genome size complexity.
Genome, what is genome? What is genome size, and what is the complexity in it? Today's class is entire we discuss what is genome. What is genome? It's a complete set of genetic material present in the organism. Means what is the haploid content of the DNA? It's present in an organism. It is called as genome. And somebody is asking about what is the haploidy in cytogenetics. Haploidy is nothing but the condition of existence of one basic set of any genome genetic material is called as an haploidy. What is genomics? Actually, who coined the term genome?
Johans. Sorry, sir, not Johans.
Winkler coined the term genome, and the genomics were coined by Roderick. Then, what is exactly total again further redefinition? If we want to further redefine the genome, the total amount of genetic material present in the haploid set of chromosome in an organism. Because for organism having haploid, diploid, monoploid, triploid, tetraploid, pentaploid, ploid is n number. Sometimes it's like octoploids. But if you want to define a genome of the potato, It's a tetraploid. You can't say total amount of the DNA present in the potato is not a genome. The haploid set of the genome is called as your haploid set of genetic material is called as a genome of an organism. You should clear with this point. Only the haploid content of the particular DNA. If it is a diploid, only haploid should be considered as the genome. Then how this genome is organized. The degree of organization and regulation of genetic material within an organism genome is called as, and understanding this is called as genomics. Then what is C value? This is one thing we need to take care. What is C value? It's the total amount of DNA in the genome. It's measured in the picograms. It's measured in the picograms. You should remember. Why I am listed all the points wise? Because these are the things you need to mug up and you should remember. Okay, it's not like concepts. That's why I given a detailed information. Then, for example, if is a there is a smallest fern, it's the biggest genome in the plant community. A small one of the grass I forgot is a fern having the largest recent discovery. It is largest genome. It's a very smaller size, but you can take a mung rice, so much big, having a nearly only just 470. It's in GBs actually. You take lentil only. As we are working, I'm working. Lentil. You know what is the genome size of lentil? It's 4.3 GB. Mung bean. Both are similar. Like when you look at the plant architecture, both look similar, same plant height, same size. Okay. But mung bean is how much? Only 430 MB. Our lab is working on both lentil and mung bean. Whenever my sir will give data of lentil, like when we do sequencing, you know how much data it will consume. So one genotype it is 4.3 GB and mung bean it's just 430 MB. But normally what we assume, the size of the genome size will increase according to this complexity of the organism. Means, for example, you take if the plant will like if the organism having a bigger size, we think that the DNA will be like genome will be the bigger size. It's not like that. It is there is no correlation between organism size or organism complexity with the genome size. This is what a paradox means. What is paradox? It's a uh, it's a not common thing. It's an uncorrelation thing. Means your genome size and your body size will not correlate. It's not correlated. This is called as paradox. Means the paradox at DNA level is called as C value paradox. Means first we should know what is C value. Means total DNA content in the haploid organism, but it is not correlated with the organism complexity. Means it's not correlated with the size of the organism. How big is the organism? It's not correlated. Even a smallest organism, like a smallest fern, may have the biggest genome. Even the biggest plant, like coconut, all those things, may having a smallest genome. There is no correlation between organism size and genome size. Understood what is C value paradox?
Yes, sir.
Yeah. Then it's more complex in the organism. We think that there is a more genetic information it's need to survive. Means we think that larger is the C value. But even a smaller, for example, this correlation is somewhat similar for some extent. Like for example, virus having a very smaller genome because it having a smaller smaller things to work. But even though it having a smaller genome, like how much function it will do. Then bacteria have smaller genomes, and eukaryotes having bigger genomes. Up to this correlation. But when we are looking at some organisms, for example, you take paramecium, like they having larger genome than the elephant. Like that, there is a uncorrelation things will be finding. We will find out that is called as C value paradox. Then, why we need to study about the genome? But exactly why we need to study the genome, genomics? What's the need? We'll study that. When I do experiments, this is the thing. First of all, we need to understand the genome of any organism to understanding the evolution. For example, how cereals were developed. For example, graminate, take cereals uh, like wheat, rice, sorghum, whatever sugar cane, these are all graminaceae, but they having different character. Someone is wheat and the rice is C3, but sorghum and maize is C4, even though they are same graminaceae. Even though they share 80% of the genome similar, but why they have the different function? This can be understood by, under, by understanding the genome of an organism. If you know the genome, okay, here there is a variation in the sequence, so that may be why is sorghum is C4, that's why rice is C3. Nowadays, people are working on how to make rice is a C4, like C4 rice. How we can do all those things when we know the evolutionary history of the between the C4 plant and C3 plants. So why the C3, why rice is lacking which genes, like, like which genes rice is lacking, so it become C3, which genes like which sorghum having which gene, sorghum having which extra gene, so that it will become a C4 plant. These things can be summarized or understood by understanding the genome of an organism. Wait, hello? Hello? One ನಿಮಿಷ. Sorry. Yeah. To understand this evolutionary histories, to understand what is the relation, phylogenetic relation between the two organisms, to understand what is the orthologous gene, what is homologous genes, how much relatedness between the organisms. If you know all those things, you should understand the genome first. How they are, how the genome is organized in an organism. It should be known. That's why understanding the genome is very important while understanding the evolution. Then then to understand the diversity, like everyone is doing diversity, like for example maize having highest diversity in the northeast India, in case of color, in case of height, in case of protein content, in case of sugar content, everywhere you find diversity, but why they are so different? To understand this, we should, if you want to find okay which genes are responsible for which trait, we should know, we should understand the diversity. How we can understand the diversity? When we know the genome sequence, or when we know how the genome is organized, then only we can understand the genetic diversity. Then we can understand, for example, disease diagnosis and treatment. For example, everybody talking about uh, gene therapy. Like if you are some genes are defective, we can correct that gene and we can make healthier individuals. If you want to correct the particular gene, you should know what is how the genome is organized. For example, in case of haploid, like in case of bacteria, no introns at all, only exons. Like you can directly mutate exons so that expression will change. But it come to eukaryotes, it having so much of introns, so much of repeated gene, if you hitting your particular gene is very difficult. You should know how much or how much amount of the DNA is repetitive, how much amount of junk DNA is present, where exactly your exon is present, how it is organized in the chromosome, how it is organized around the histone proteins. You should know all those things. Then only you can make all the therapeutics, gene editing, what are the things you are do. Before understanding the genome, you can't do anything. Then for crop improvement, because what QTL mapping, GWAS, whatever you will do, you're understanding the genome, then only we can able to do crop improvement. Then epigenetics. So many will talk, like we talk every day what is epigenetics. Okay, it's a very hot trending topic. You know what is epigenetics? Can anyone define what is epigenetics? Oh, I'm audible or not? Hello? Hello?
Yes, sir.
Yeah, because I'm going like, okay, because today I'm not asking many questions. What is epigenetics?
Sir, studying about the histone proteins and the materials that are
What is
involved in
epigenetics? Just one line definition.
Changing genotype without altering in sequence of DNA.
Exactly, this definition. See, epi means other than the change in the sequence of the DNA, but there will be a change in the phenotype, and that change is heritable. You should define in this way. Okay, this is called as an epigenetic. If you want to know about epigenetics, you don't know how your particular DNA is there in the cell. How DNA is where DNA is present, you don't know, and how it is organized in the cell, we don't know. Means how we can able to understand epigenetics. To understand the epigenetics or mechanisms, whatever it may be, we should know the how the genome is organized in the cell. Okay. Then another thing is factor affecting the genome size and complexity, repetitive DNA. For example, if you take eukaryotic gene, it's a very simple. All 90% is a unique DNA. But if you take eukaryotes, or I take maize, 70% is repetitive DNA. And that too have more number of transposon elements. I will discuss that thing. Means, you see, how much amount of repetitive DNA present in the genome is also determines your genome size. What is the genome size of maize? Can anyone? I think it is 2500 MB. Should know at least 5 gram. What is the genome size of wheat? Even though I am reading many times, every day I will read the books because my target is something. If you want to reach the target, you will work a lot. Okay? Wheat, what is the genome size of rice at least?
250 base pairs, megabase
Is it
pairs,
so?
250 megabase pairs.
Yeah, I think it's 400 plus. Okay, you check it. Then what is the genome size of Arabidopsis? At least you should know model organism. What is the genome size of
Nine,
C. elegans?
sir, Arabidopsis 99 megabase pairs,
Exactly.
100.
It's 100 MB. Okay. Like this. And what is the model organisms present?
Sir,
What is model organism?
Arabidopsis and frog species.
In case of plants, it's Arabidopsis. In case of crop, is rice. Okay. Rice is in case of crop. Arabidopsis in case of plant. At least you should remember this. Arabidopsis in case of cereals. And again, wheat. Should remember rice, wheat, maize, and maize. Maize is a very important in case of this is called a Cinderella of genetics. Okay, you should remember at least these four crop: rice, wheat, maize, are okay. These things are very must. And how much percent of repetitive DNA? How many number of genes present in Arabidopsis? This is what this class. So Okay, leave it. What is repeated DNA? If you don't know this, you're not going to understand entire marker concept.
DNA base pairs repeating, sir, in case microsatellite model or macrosatellite model.
Yeah, exactly. Okay. For example, this 8 is repeated 1, 2, 3, 4, 4 times in individual 1. This 80 is repeated 1, 2, 3, 4, 5, 6, 7 times in individual 2. 7, 8. Here it is repeated 9 times, 7 times, it is 4 times, individual 3. See, this particular nucleotide sequence 80 is repeated 4 times. Means it's a repetitive gene. It's a microsatellites. Means like some like 1 to 10 base pairs repeated nearly 10 to 100 times. Okay, 1 to 10 base pairs repeated 10 to 100 times. This is called micro. If it is 1000 times to some MBs, like 1000 base pairs repeated several million base pairs, several MBs, it is called as mini satellites. Many satellites very larger in the size. Microsatellites are very smaller, like 1 to 10 nucleotides repeat nearly 100 times, 10 times, 12 times, like this. They are microsatellites. These are repetitive, okay? Highly repetitive, moderately repetitive, large, highly repetitive. These things you should, types of repetitive is very important. Types of repetitive DNA, and at centromere, what type of repetitive DNA is present? At telomeres, what type of repetitive genome is present? At X chromosome, like inactivation of X chromosome, why it happens? Because of most of the genes will fall under repetitive DNA. What is euchromatic? What is heterochromatic? Okay, you should remember all the things. Means how much amount of repetitive DNA is present in the genome, it depends your genome size. Then the polyploid. More the number of polyploidy, larger will be the genome size. Take wheat, which is hexaploid. Hexaploid, sorry, 2n is okay, 6x. Hexaploid, you know what is the genome size of wheat? I think 1.8 GB, I think. 1.8 GB, yes, 1.8 GB or 18 GB.
13, I think so.
No, I think. Actually, in one paper, it will be like that. It's nearly 1800 MB, I read. No, no, 18 GB. Some GBs, it's more than 10 GB. Like, okay, you just search it. Like 1.8 GB, 18 GB nearly. Like this, if the more the polyploidy and more will be the your genome size. Then gene duplication. For example, what is gene duplication? Can anyone define? What is gene duplication? What is gene duplication? I'm searching genome of wheat. Other I have to confirm. Please tell the answer. Yeah, it is 18 GB, like 14.5 to 16 GB. No, 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 17 GB, 17 to 18 GB in one paper. Actually, why this difference will be arises? Because every day, wheat genome is not a complete sequence. It's a draft. Every day, like new paper will come, they will announce a new gene. And somebody is using spring wheat, somebody is using soft wheat, somebody using another type or like winter wheats, and somebody using different genotypes. That's why it is vary. Means you should just remember 14 to 18 GB it's varying. Okay, because it's having A, B, D genome, and there is so much modification in A genome, B genome, D genome across the different species. Means a lot of variation, lot of size variation also there. It depends on which species he is using for sequencing. That depends. Okay, that's why this variation. Even rice in some paper you take 380 MB, in some paper 430 MB, in some paper 410 MB. Why this variation? Because if somebody using Vareza indica, normally we are using Vareza japonica, Nippon Bore. Okay, this depends on which species he is using. That's why, but you should remember average. Means it is 10 plus GB wheat. Rice only just 4 and 300 to 500 MB. Mung bean 300 to 400 MB. Lentil it's 1.4 like 3.4 GB. Like this you should remember. Arab gas is only 9900. Okay. Then say celiac acid, just 100 MB. Like this, you should remember. What is gene duplication? Can anyone tell? How many number of times? Gene families, can anybody heard about gene families, multi-gene families, super gene families? Not read for JRF. Then how are you going to understand very complex things? If I teach very complex things, it's very difficult to understand. Should know how gene will duplicate, how new genes will arise, what is the different cause of the arises of the new genes because of duplication, what is gene families, what is multi-gene families, what is super gene families, what are the examples, globulins, hemoglobulins, globulin gene family. Okay, these things are basic things. Okay, should read. Because of gene duplication, if more the duplication, more will be the genome size, more will be the complexity. See, if the bacteria no duplication, that's why it's a very simple. And genome reduction. What is genome reduction? Most of you take progenitor species, they are all polyploids. But nowadays, cultivated species you take, most of the cultivated species, they are diploids. Because during evolution, there is a reduction in the genome. Reduction in the genome, and there will be a split in the polyploids. It's also one of the major cause of speciation. And genome reduction, how much genome reduction is also determine the genome size. Larger will be the size of genome reduction, smaller will be the size of the genome. Like that, you should remember. It's also one of the factors. And other environmental factors because of like epigenetic modifications. Okay, because of stress, some epigenetic modification will happen. Maybe your gene will become compact. It's not available for any replication, transcription, translation. If it is present in the no methylation markers, it will become very active and your gene will become expressed during evolution. Maybe there will be a change in the gene, like a change in the gene sequence due to mutation, all those things. These things you should remember. One is how much amount of repetitive DNA present, how much amount of polyploidy, how much amount of DNA duplication, how much amount of genome reductions, and what are the environmental factors which all affects your genome size of a particular organism. Then we can compare, like if you compare different genome sizes of the organism, bacteria, it's less than 500 kb. For example, you take Mycoplasma genitalium with only 500 genes and 580 kb. It's a small fungus. And it takes organism cellulosidum by having a 13 megabase. See how much? Only 13 megabases. It's one of the but you can take eukaryotic microbes. It having some 100 billion base pairs also possible. For example, you take Saccharomyces cerevisiae, it having 12 million base pairs. These are some model organisms regularly using in the field of genetics actually. Amoeba dubia having like it's one of the eukaryotic bacteria I think. It's a 200 billion base pair. And when you take the plants, for example, Lollipop pine tree have a genome size of 20 billion base pairs. You should remember all these examples. Paris japonica plant has genome size of 150 billion base pairs. See how much big the genome size. One thing when you observe the pattern, see prokaryotes having very small genome. Eukaryotes, microbes, microorganisms like fungus, they having little bit bigger genome than the bacteria, but the plants having more more the genome size. But even though some correlation was there, but when you take some exceptions, it will uncorrelate. It's called as p-value paradox. Like this is how what I explained. Like for example, this is the genome size of the different organisms. In any news, like in any paper, you will get it. Please at least remember the genome size of the model organism. Then, if you look at the maize as some example, see it having 2.5 billion base pairs, means it having nearly like 2500 MB or 2.5 GB. Then see 85% of the genome is repetitive. I told 70%, like 70 to 80% of the genome is repetitive. Only hardly 15 to 20% is your the genes which are coding for the protein. With that 20% only, how much variation is present? And out of that, 90% is transposable elements. And there is a presence of large number of copy number variation. Even only 20% of exons is there. That 20% exons among this across the different species is highly variable. This is what the things we need to look when you look at the genome organization of the particular crop. Like here, first you should look what is the genome size, then how much amount of repeated DNA, how much amount of unique DNA, how much amount of transposable element, how much number of copy number variation present in the particular species. This is what generally we look for the genome organization. When you say, okay, genome organization of particular crop, at least you can able to say this. What is the genome size? What is ploidy number? How much amount of repeated DNA? How much number of transposable elements? How much number of unique reads? Unique genes, and how many number of genes present in the particular species. For rice, I think hardly 20,000 genes present. Arabidopsis, nearly 20,000 genes present. Sorry, rice, I think 40,000. Please correct it. Okay. This statistics you should know when somebody asks you, okay, what is the genome organization of particular crop? And everybody understood what I am going to say? You're getting or not? These are the basic things. What is genome?
Sir, I have a doubt regarding this, sir.
Yeah,
Sir, if we are isolating a DNA from a plant part, for example, leaf,
yeah,
whether it will have 2n number of genes, sir, isn't it, sir? 2n 2n genome, sir, rise if we isolate
yeah,
the DNA from the leaf. But
yeah.
when we sequence it, we will sequence the each of the genome twice, sir. Then
No, no. Yeah, we can sequence only one single haploid genome.
yes, sir. How is that possible, sir?
See, for example, chromosome. Chromosome set will be there now. The DNA, we are taking only the DNA when you are mapping. For example, you are isolating the DNA. Your doubt is it's 2n, but how the gene will come dn? See, while we are sequencing, you are isolating the DNA, and the DNA will come from both the ploidy. Okay, but while doing the alignment to the reference genome, it's already present reference genome. While doing an alignment, you align the reads. Okay, then you will design the sequence.
Okay, sir.
Okay, which are not like they will align twice. They will again, like for example, they having same locus. The reads will align same position.
Yes, sir. Yes.
We are isolating the DNA. Okay, DNA will be content may be different. For example, haploid DNA having 10 picograms, the diploid having 20 picograms. But while aligning the locus, maybe it will align 2 times.
Yes, sir. Okay, sir.
Yeah, it's not like okay. We are looking at the DNA level, but we are normally looking chromosome level, like 2 chromosomes. But we'll do sorting. In I think you know about chromosome sorting, where to separate out A genome, B genome, D genome. We'll use chromosome sorting also.
Okay, sir.
Chromosome sorting also. Well, case of it diploid is no problem. It will align same. But if the different genome is there, like A, B, D, we have to do separately. First, you isolate A genome, B genome, D genome, then sequencing separately. Okay. But if
Yes,
your
sir.
A
This.
is, they will align it the same. They will align
Okay.
2 times. That's not a problem. But if you are
Yes.
having different genomes, you should do chromosome sorting. Then you should do sequencing of separate A, separate B, separate D. That's why every paper you see, chromosome sorting will be there. Every wheat paper you see, genome sequencing of wheat paper, there will be a one term called as chromosome sorting. Your doubt is correct.
Yes, sir. For a long time, I
The
have been.
same genome repeated how many number of times, it's not bothers your sequencing. But if you have different genomes, you have to do separately. Your doubt is right. Yeah, any doubts? I'm happy someone is getting. Then there are types of gene density. What is gene density? How many number of genes present in the particular genome? Maybe 20,000, 10,000, 40,000, this thousand. It is called as, and again, if larger is the genome size, more will be the gene number. This statement is correct.
No, sir. Not correct, sir.
There is no correlation between these also. It is called as G value paradox. The black and red combination is very nice. G value paradox. Understood? There is no relation between the genome size and the number of genes. It is called as G value paradox. This is the one type of complexity. And C value paradox I told already. Repetitive elements. Alternative splicing. This is also another factor. For example, gene A and genotype. Here having 20,000 genes. One species having 30,000 genes. But it may having 40,000 protein. It may having only 30,000. Is any case it can ever it is possible or not?
Possible, sir.
Yeah, because of alternative. Actually, I should have asked this question before this. Yeah. Why? Even the 20,000 genes may code for 40,000 protein. You can see protein number is more than that of gene number because of alternative splicing. It's not like that if the genome having only 20,000 genes, it will only code for 20,000 proteins. It's not like that. Even the genome having 20,000 genes, it may code for 40,000 protein also. It's possible because of alternative splicing. This complexity also arises. Then non-coding RNA. For example, if the 20 out of 20,000 genes, it will coding only 10,000 proteins. This is also possible. How? Out of 10,000, it is non-coding RNA. It will not going to express as a protein. This is also possible or not?
Possible, sir.
It is also possible because of non-coding RNAs. Then the genome organization. For example, if the more number of genes near to heterochromatic region or more methylation around this gene. But in other species, there is no methylation. It will express, it will give protein. But it will, because of methylation, it will not express, it will not give protein. This organization of particular gene is also matters to expression. That's why complexity arises at organization level. Okay, what level complexity arises? Understood? At gene number level, there is a complexity. Because of repetitive genome, there is a complexity. Because of alternative splicing, there is a complexity. Because of non-coding RNA, there will be a complexity. Because of the where based on the methylation pattern, where the particular gene and genome are present, uh, it is also one of the complexity. Then I think it's a repetition. This is then how to analyze. There are methods to study how we can study the genome organization. One is you can directly sequence the DNA nowadays. We can get information of genes, what are the regulatory elements, the structural annotation. By using structural annotation and functional annotation, after sequencing, we can able to know understand the genome organization. Then one of this great example is called as Human Genome Project. When Human Genome Project is started, can anyone tell when Genome Project is started?
1990.
Yeah, exactly. 1991. I think. Then it's completed nearly 2003. The complete sequence of human genome was sequenced. Then how many? What is the genome size of human being? See, these things are very important. 3 billion base pairs. Which is the largest chromosome, which is smallest chromosome. These things, human genome project, at least one or two question definitely will come in any other exam. You should read about human genome project. Then we can by using fish and dish, we can able to study fluorescence in situ hybridization, genomic in situ hybridization. We can able to study how much amount of repetitive DNA is there, how much amount unique like C banding, R banding. These are the banding technique we can use. These are the fluorescence technique we can use. We can understand the genome by using this technique. And Kihara is one of the important to study the genome analysis. We will discuss what is exactly genome analysis while discussing cytogenetics. These things, what I want to discuss, accidentally I taken this class. Now you have an idea what is genome, what is genome size, what is repetitive DNA, at what level complexity arises, what is C-value paradox. Understood? Any doubts up to here?
No, sir. Understood, sir. Very well.
Pavitra, understood? Nitish? Nitish Singh,
Yes, sir.
understood?
Yes, sir. Yes, sir.
I'm going faster. Okay, it's optimum level. Manikarnika,
Yes, sir.
Lohit,
Yes, sir. Understood, sir.
I will discuss some of the because I have time, like 15 minutes. What is we discuss what is marker and everything. I will not repeat. Soon after the discovery of the DNA, scientists started thinking about what is there in this DNA. Okay, when that experiment, while conducting that experiment, the significant achievement was done by Bertson et al. This paper was published in 1980 in the some human genetic journal. Uh, I I given the reference. You can read this paper. He said that signifies that a single restriction enzyme generates fragments differing in the length from the same genomic regions of a different individuals or strains or lines of a given species or a different related species. Means there is a variation in the homologous fragments of the length of the DNA is known as Rflp. I think nobody has got what I have read. Will explain. For example, if A is there and B is there. Here, what is the restriction site of E. coli? I forgot, and that's why I'm asking. You just take this is one of the restriction site. Here. Think this is A individual and this is B individual. And this is the same locus. Think this is from like 10 base pair to 15 base pair, 16 base pairs on same location. 10 base pair to 16 base pair. They are in the same location. But there is a point mutation in the restrictions. Instead of T, here G is there. Means if there is this type of point mutation, for example, you're treating these individuals with E. coli enzyme. It's one type of restriction enzyme. We will discuss what is this E. coli enzyme. E. coli can able to cut this particular DNA into two pieces because it finding its restriction sequence. But here, due to mutation in this particular allele, the recognition of the sequence will be lost. Means your E. coli unable to cut here. Unable to cut here. Means here you are getting only
A
as
larger
its band is
fragment.
intact. There is no what?
Sir, I came to tell a larger fragment.
Same. It's not like larger, it not become smaller. What is the size is there 10 to 16? It will be like 10 to 16 only. But here it will become 3. It is cutted here, it is cutted here, it become 2 base pair. Another it is 4 base pair. But here it is 6 base pair only. The size will not going to change because nothing is happening. Here, due to this restriction enzyme, cuts this sequence into 2 fragments because it finds its recognition sequence. But due to point mutation or anything, insertion, deletion, whatever it may be, here your restriction enzyme unable to act on this particular sequence. That's why you're getting nothing. Like you're getting same length as like original sequence. See, A and B individual can able to differentiate by this point mutation by using the restriction enzymes. Again, you read the definition. Means a single restriction enzyme which generates generates fragments. These are the fragments differing in the length. They are see here 6 base pairs, but here I have 2 base pairs and 4 base pairs. There is a difference in the length of the fragments after your treatment with the restriction enzyme and from the same locus. They are from the same locus. But although it is given, there is a different length of a fragment due to point mutation. This is called as Rflp. Means restriction will generate, means restriction enzyme will generate fragment length polymorphism. I told what is polymorphism. Means there is a difference. The difference is due to difference in the length of the fragment. See, here you are getting smaller fragments, but in other case you are getting larger fragment. Same fragment as like in uh, the original sequence. There is no restriction. Okay, you can able to differentiate these two individual by just treating ecoenzyme or not.
Yes, sir.
Yes, sir. No. Yeah. This is itself is called as restriction, fragment, length, polymorphism discovered by Birds. How we can exploit? What is restriction enzyme? Again, a question. Yeah. I think many of you know what is restriction enzyme. Restriction endonuclease. How many types is present? Type 1, type 2, type 3. Only type 2 is used in the biotechnological experiments. Type 1, type 3 cannot be used. Why it is so?
It might create blunt ends.
Another reason?
Sir, it uses only one cofactor, magnesium. ion.
Wait, actually. So, can you repeat your answer? I was just looking some. What is type 2? Why type 1, type 3? See, these are the basic things. Okay. UG biotechnology. You forget on all the things. Okay. If you forget all the things, if you want to learn extra things, it is very difficult. You should not forget the basics. What is restriction enzymes? What are the uses? What is the use in the biotechnology? Without knowing restriction enzymes, you do nothing. Like you can't do anything. What is cloning? Cloning is based on restriction enzymes only. And whatever you do without restriction enzymes, it's nothing is possible in molecular biotechnology. Just please read. And Rflp is the first molecular marker to be developed, and it's based on southern blot hybridization. I will discuss southern blot hybridization. I will not ask the question. And it's a codominant. The Rflp marker is codominant in nature. I will discuss how we can explain. And this marker system is highly reproducible and very robust. And it requires high quantity of quality DNA, and it uses radioisotopes. It's like these are the advantages. These are some of the limitation of this marker system. These are the advantages, and these are the limitations. Means if you do, if you are using this marker system, we need very pure DNA, because if DNA is contaminated, your restriction enzyme can unable to uh, take enzymatic reactions. Means you should have maintain purity, you should maintain optimum temperature, like that. See, what is the optimum temperature for enzyme activity? All the things you should read. This is available in the Genome 3 book. You're not reading all those things. Genome 3. Who wrote the Genome 3 book? Primo's book. Please read all those things. Okay, it's very important. Then, yeah. Understood all the points you got from this slide? Anybody have doubts?
No, sir.
No,
No
sir.
answer.
No doubts, huh?
Yes, sir. No doubts.
What is reproducibility and robustness?
Sir, if you are genotyping multiple samples, we should able to genotype multiple samples. That will be called as robustness, sir. Reproducible means same results we need to get, sir.
Can you explain some elaborate so that everyone can understand this point?
Sir, if we are having
Satya only, no?
No, sir, I am Venkat, sir. Jai Venkat.
Okay, Venkat, okay.
If we are having some 100 number of samples and we are genotyping for a Rflp marker for a particular using a particular restriction enzyme, it should be producing some results, sir. That
Okay.
will be reproducible, sir. And robust means it should be able to we should be able to genotype a large number of samples, sir.
Okay. Yeah, somewhat you correct. For example, your lab A. For example, my lab, Prem Kumar's lab, you are doing experiment with the same set of genotypes. For example, you are having 10 genotypes. You are using Rflp marker. You are getting some results. In Venkat lab also, same 10 genotypes, same marker system, you should get same results. And if you do any number of times, you should get same number of results. That is called as reproducibility. And this marker system is highly reproducible. Then robustness, as you said, it's right. You can screen large number of genotypes, but when compared to other marker systems, then advanced than this, they're less robustness because it involves southern hybridization. Southern hybridization itself is a very, very difficult task. Then what is codominant nature?
It should be able to differentiate between homozygote and heterozygote, sir.
Exactly. The marker system should able to differentiate. For example, this is one system. This is what is this system called? First system. First condition, you are not going to observe any band in the recessive homozygote, but you are observing same band in the AA and capital X small a, heterozygous and homozygote. Means this is dominant system. You can't able to differentiate capital A, capital N, capital A, small a. Means you are getting the same bands here and no band in the recessive. Means it having a dominant marker system. Then, if it is a codominant marker system, you are getting here 50 base per band, you are 40 base per band. Here two bands you are getting another band. Here like 40 base pair. I will show the diagram. Here both 50 base pair and 40 base pair will be present. Here see, because of this nature, it can able to identify capital A which one, capital A small a which one, small a which one. This is called as codominant nature. It's not like that. It means you are getting the allelic representation from the both the alleles. Capital A also having specific band, small a having also specific band. When they came together, the both the band present. And here small a specific band will present. Here capital A specific band will be present. This is why it's a codominant system. But in case of dominant system, in capital A condition, you are getting same band as like in the heterozygous also you are getting same band. But in recessive, you will not get any bands. This is called as dominant system. And there are several markers are example for dominant and several markers are codominant. Like SSR, Rflp, there are codominant like SNP. But the dominant is RAPD, ISSR, scar marker. They are dominant system marker. Understood what is marker codominance nature?
Yes, sir.
Yes, sir.
Then, and it's based on southern hybridization because if you are treating a restriction engine, means you should go with southern hybridization procedure. What is southern blot experiments? Very ex- First, how to do RFLP? So, for example, your guide told you use RFLP markers and do genotype. How you will proceed? First, you should isolate the DNA. Then you should treat the DNA with restriction enzyme. Then you should do gel electrophoresis. Then you should shift all the DNA from gel to blot. Then you should go with hybridization with probe. Then you should wash. Then you should use detection system. Like the auto radiography system will be there. We are using that. I think many of you have an idea about Southern blotting technique, but I will discuss. First, you should isolate the genomic DNA. Then you should treat with the restriction enzymes, like small fragments will come. Then you should load this DNA into fragments, into wells. Then you have to run the gel. Then band will look like this. For example, here take how many, here some kb, here some kb, here some kb. Because you're treating with restriction enzymes, your fragmentation will happen. It's not your genome having many restriction sites. It's not like that only one restriction site is not there. You maybe for every probability will be very high. For example, if having 6 base pairs, probability will be very high having restriction sites. There is some formula I forgotten actually. What is the probability of the restriction site for a genome? There is a formula actually. There are so many restriction sites, so many fragments are there. Then you should do Southern blotting. You should shift all the DNA present in the gel to the nitrocellulose membrane. Then you should hybridize with the radioactive probe. I hope everyone understood what is probe. Then you should wash. Then you should fix this into the cell. Then you should go for the autoradiography. Then you will visualize where your DNA, which location, which locus your variation is present, you are going to get. This is the overall protocol if you are running an Rflp marker. For example, here the band will be there. How we can do blotting means you keep the gel here, then you should put some filter papers, then you should put some weight. All the gel will go to nitrocellular membrane. Then you will get after auto radiography, you will get images like this. See these bands. Understood? How what is Southern hybridization? This I took from
Yes.
Primo's book. It's a very beautiful book. Please read it.
Yes, sir. Understood, sir. Sir, one small doubt, sir.
Yeah.
Sir, we are using this hybridization with a probe just only to easy genotyping, isn't it, sir?
No, see, I
Because
told there are
there will
so
be,
many.
yes, yes,
See,
sir. There will be
there are
lot of,
so many restriction sites. If you're treating restriction engine, see, if you treat, see how much cutting happens. Means, for example, this is a large genome. Here on restriction site, here on restriction site, here on restriction site, here on restriction site, here on restriction site is there. Means you're getting large number of fragments. And you are want one locus, particular locus. Oh, I want this locus. I will design this probe. And if probe is, where the probe is in, how probe, that's why I asked. I hope you understood what is the probe concept. Probes are known sequence. You know this probe at which location of the genome, you know the address of this probe, where it is present on the genome. If this probe will bind to any fragment, okay, you can say this, okay, this fragment is from that location only. Understood?
Yes, sir.
The
Yes, sir.
local specific. That's why you're using probe. That's why we are doing southern hybridization. Okay. Okay. You can say this. That's why RFLP is locus specific marker.
Okay,
Means
sir.
we should know. We know the locus where, based on proto hybridization, we can able to say, okay, this variation is there in the particular position of the genome. That's why RFLP is a very robust in nature. That's why you're getting reproducible results. You can able to see, okay, this location only our variation is present. Understood?
Yes, sir.
While stopping the stage. Okay, tomorrow we will discuss what is diversity array technology. It's very easy. Then what is single feature polymorphism. Then how to isolate DNA and these things we will discuss tomorrow. Okay. Okay, any doubts up to this? When testing in the lab, should we give same result? Yeah. Thank you. Any doubts up to this? Shall I end the class?
Yes, sir. Thank you, sir.
Thank you,
Please
sir.
read. Actually, it seems like everyone is very poor in the basics, especially regarding genomes. For this, you read a genomes chapter to get the basic understanding of these things. Like genomes book, genome 3, there will be black color page will be there, written by Robert Brown. Okay. Bye. Good night.